Okay, class. So we wanted to cover this, these questions. We had these free response questions. I told you before that that the um, signaling transduction pathway video that we watched and sort of analyzed by Bozeman is a good example of the type of questions that you might see regarding this content right here, this topic of cell signaling and signal transduction. So this is just a more specific example, and you remember in the case in the video that we watched, we were actually looking at epinephrine as being the messenger or the signal here to this G protein, or this protein embedded in the cell membrane. And so a similar thing is happening. You have this sort of cascading effect where you have, the ligand attaching to this original protein, and then the G protein, a subunit, coming over here to this other enzyme to activate it. And then there's another effect via CAMP and protein kinase, which will eventually phosphorylize, and you should remember that term, phosphorylize, or activate this enzyme phosphorylase at the bottom, which then will facilitate the breakdown of glucose. All right, so if you have any, still have any questions about this process, it would be good to review this one. This is video 38, signal transduction pathways from Bozeman. But returning back to this, we'll see here that we have basically, if we read through this thing, read each question carefully, write your response in the space provided. You can't use outlines, bulleted lists, or diagrams alone. They're not acceptable. You can use them in addition to your, basically written out in sentence form answer. And in this case, all they did was, use growth hormone and insulin. These are two protein hormones that regulate carbohydrate metabolism, meaning the process of breaking down carbohydrates. And this occurs in hepatocytes, the liver cells, through that sort of same signaling pathway. Something's going to happen, from a ligand, and then there's going to be sort of this continuation of reaction, or series of reactions to produce a cellular response. And it says researchers investigated whether the two hormones trigger the same or different intracellular signaling pathways in hepatocytes. So, just scientific experimentation, controlled experiment. Basically, they're using cultures of hepatocytes, and in one, they pretty much observed the phosphorylation or activation of STAT5. And then in the other, in B, they observed the phosphorylation of protein kinase B. All right. And so remember, just think about protein kinase B in this other experiment here, or protein kinase, actually. When we looked at this one, remember that protein kinase at some point here was activated through CAMP. The regulatory portion was detached from the catalytic portion, which then the catalytic portion was able to come down and with some energy from ATP actually activate phosphorylase, which then would come over here and facilitate the breakdown of glucose. So and at this point, we're not worried about what STAT5 is going to do. We just want to see, we're looking at the levels of STAT5 phosphorylation and the levels of protein kinase B phosphorylation. And we can see here, just to take a good look at the data on this one, we see a couple of things we want to observe is that from 0 to about 10 minutes, they're saying, and if you read the questions, you'll see that after the ligand, right, that's the first question, activates or initiates this signal transduction pathway, is about 10 minutes. And we see that reaches the maximum level of phosphorylation for STAT5. And at about 30 minutes, there's this sort of drop at about 30 minutes. So within 10 minutes, we reach maximum effect or phosphorylation in the presence of growth hormone, right? So for STAT5, growth hormone is causing this single transduction pathway. Little effect, if any, due to insulin on STAT5. On this one, we see that insulin has the phosphorylating effect on protein kinase B, and we see at probably around 30 minutes it reaches its maximum, and then pretty much, it's still at a high level up to about, let's say, 80% here. Okay, so the first thing you do, you want to analyze the table, and we'll see that this went for, they ran this experiment for 240 minutes. Okay, so for about 4 hours to see, the lasting effect of growth hormone and insulin on these signaling pathways, right? And so we come down and it says, hey, describe the first interaction that triggers signaling to begin in a target cell. What they're looking for is for you to say something about a ligand connecting with or docking with a protein in the cell membrane. Okay, that's it. Now let's see. I'm going to show you that here in the suggested answers or the scoring guidelines. You see this thing says scoring guidelines here. So if we talk about part A, we'll see it says the response indicates that the ligand or chemical messenger, in this instance a hormone, either insulin or growth hormone, binds to a receptor protein in a target cell. That's all they're looking for. Okay, and you'll see here, okay, there's 10,000 words, but if you know what we're talking about, then you pretty much, you have covered it in basically one sentence. And then B, it says identify a dependent variable in this experimental design. Identify negative control for the effects of hormone addition, and identify a likely reason why the researchers continue their experiment for 240 minutes. So if we look at that, we'll say, okay, identify a dependent variable. Let's go back to what we know. Up here, dependent variables are the things that we are observing, right? So you can say in general, we are observing the levels of, phosphorylated proteins, right? Or you can say specifically levels of phosphorylated STAT5 and levels of phosphorylated protein kinase B. All right, and what else are they looking for? A negative control. So we would think here, we would stop and think here now, if we were to do this experiment and we wanted a control group, and we wanted to figure out a scenario that would produce no results. Okay, would have no effect on STAT5 or no effect phosphorylation or no effect on protein kinase B phosphorylation. Well, we could not add any growth hormone or insulin. We could add a defective growth hormone or insulin. When these things are protein hormones, we could do that. But let's see what they say here. It says, so we're trying, a negative control is a way to get no result. Like, we're acting as a control. So it says the response indicates that a dependent variable is the level of phosphorylated signaling protein, or the level of phosphorylated STAT5, or the level of phosphorylated protein. So we answered that one. Then we go to the next one. And your response would indicate that a negative control for the effects of hormone addition is the 0 time point when the hormone is added. So we know that nothing from these two data at point 0, nothing is happening. At time 0, nothing is happening. So that's where we would expect nothing to be going on. Or adding no hormone, or even adding a denatured hormone to, an identical culture of hepatocytes, right? And, taking samples at the same time. So negative controls are useful because basically you want to have a group that has not received anything, and watch and compare, in this case, those levels of phosphorylation of STAT5 or protein kinase B to these two experimental groups that we have. And then, let's see. All right, so we move on and we get down to C. It says, describe the hormone and signaling protein combination that produced the greatest and most prolonged response. For the phosphorylated STAT5 response that reached 100% at 10 minutes in figure 1A, calculate the rate of decrease, percent decrease per minute in the detected level of phosphorylated STAT5 from 10 to 30 minutes. So we're going to describe the hormone and signaling protein combination that produced the greatest and most prolonged response. And if we look here, we see, okay, look here, we reached maximum, so we want the greatest and prolonged. So here we got insulin, the addition of insulin to this hepatocyte culture actually reached its maximum amount of phosphorylation of protein kinase B, and then it extended here throughout the 240 minutes, 4 hours, still at pretty high levels. Those levels didn't change, so this effect was pretty long lasting here. So I would say we probably should go with this one here. And then when we want to determine, so then they ask us to determine, the percentage for a particular time frame for the level of phosphorylated STAT5. Well, we'll say this first black dot is 10 minutes. It's at 100%. And after 30 minutes, it's at about, we'll say 35% here. Okay, so we have the initial rate at 100, or percentage. We have the percentage after the 30 minutes at about 35 right there. So we'll look at that and we'll say, okay, the rate changed about 65%. And then we need to divide that by the time it took to do that. So from 10 to 30 minutes is 20 minutes. So we'll say, 65, let's see, 65 divided by 20. So 65% divided by 20. Let's see. 65 divided by, it gives us something like, it gives us a number of 3.25. So, you can say 3.2 or 3.3. Let's see what they, which part is that? We'll go here. All right, it says, so we'll deal with the first thing. It says the response indicate that the response for the hormone might take some time to start, or that the researchers might have wanted to see how long a response initiated. Let me go back and make sure we're the right one. We did negative control. Okay, so that's part B3. Let me make sure we covered that. Identify a likely reason why the researchers continue their experiment for 240 minutes. Well, that would be, that would be because they wanted to see the extent or how long the protein hormones would have a phosphorylating effect on STAT5 and protein kinase B. And so here we get in C, describe the hormone and signaling protein combination that produced the greatest effect. That's what we're trying to look at. We'll go to C. And the response indicates that the addition of insulin caused a high level of phosphorylated protein kinase B. Yeah, pretty much about 80% of maximum right to the end of the experiment at 240 minutes. So for this question here, describe that hormone and signaling protein combination. It's, insulin on protein kinase B phosphorylation. Sorry, maybe I'm moving too fast. On this one, protein kinase B phosphorylation. And then calculating that STAT5 response that reached 100% at 10 minutes, and the rate, 1A, calculate the rate of decrease, percent decrease per minute All right, and so that's what I'm saying here for stat 5. We were at 100. We went down to about 35. I wouldn't